Hello guys, hello everybody. Welcome to my YT live streaming. This is again, I am a singer, eternal voice. Hello everybody. Hello world. Hello Philippines. Welcome to my YT channel. Again, this is eternal voice doing another moment of or another session of live streaming. Okay. Yesterday we discussed about cellular respiration. Okay, so there are two types, right? The aerobic process and the anaerobic process. Okay. The aerobic process of cellular respiration involves the presence of oxygen. Okay. And the anaerobic process of cellular respiration does not involve the presence of oxygen. See, we are now finished for the cellular respiration through the presence of oxygen, right? So there are at least four processes for that. It starts with glycolysis, and then after that, it will produce pyruvate, and it will be oxidized to acetyl CoA, and then after that, it will go to the citric acid cycle, which is known the Krebs cycle, and then it will go now to the electron transport chain, which is specifically the oxidative phosphorylation. I don't have to explain furthermore each process because I discussed that yesterday, right? So now we are focusing now to the anaerobic process of cellular respiration. Now, first, I will give you the overview for that. Okay, I will give you. Okay, so meaning to say, so this is very helpful in doing excessive exercises. Okay, when you do the cardio exercises, for example, jogging, biking, running, like that. Okay, and excessive workout to the gym, weightlifting. Okay, those are examples of aerobic process. Though at first, initially, okay, you will develop aerobic process because there's a presence of oxygen. Then, as long as you did that severely and excessively, so the oxygen will be reduced slowly and gradually, and now it will be converted now to anaerobic process. Okay, something like that. So it will change now from aerobic to anaerobic process. So since the end point or the final stage is an anaerobic process, cellular respiration, so we can categorically say that it is under with anaerobic process of cellular respiration. Okay, so to give you a clue for that, the end product is usually a lactic acid and a lactate. Okay, the lactic acid is okay, and the lactate is very dangerous to the health. Okay, so I'll discuss you further for this also. These two components. Actually, this lactate comes from the lactic acid, okay, the conjugate base of lactic acid, meaning it is the base from lactic acid. Okay, when we add water, something like that, right? So, so in the reaction of salt and water. Okay, salt and water reaction to produce this lactic acid. Okay, plus the base. Okay, so with OH. Okay, it is a base, right? Since this is organic chemistry, so usually we are referring to the OH, alcohol. Okay, plus the carboxylic acid. Okay. When we mean organic chemistry, it's always carboxylic acid we refer to, huh? It's not the ordinary acid because the carboxylic acid contains carbon. That's the only difference, contains carbon and oxygen. But in organic chemistry, not necessarily to contain carbon and oxygen. That's usually H positive one proton plus the polyatomic ion that ends with ATE, as I discussed that over and over again, right? Chromate plus H proton from water is equals to chromic acid. It's always water will be added to that. Okay, like that. So it will result to acid. Okay, like that. So it's different, totally different, really. Okay, the H positive proton. Okay, and then the carboxylic acid is CCOOH. COOH. Okay, so that's an identification. Do not be confused for that. Okay, since we are dealing with cellular respiration, so this is under biochemistry, and most of the chemical reaction are organic compounds in the biochemical reaction. Okay, so this is biochemical reaction. because we incorporate biology in the discussion. Since we are dealing with cellular respiration, meaning cells, we cannot live without the cells. Okay, we cannot live without the cells, the tissues, the organs, like that. We have millions of tissues, we have millions of cells, and then it will be developed to become an organ, something like that. And all of our organs, main organs of our body, the liver, the brain, the heart, the pancreas, the kidney, okay, the reproductive organ, the endocrine gland, the pancreatic gland, the pancreas, something like that, okay, and the gut, GUT, small and large intestines, okay, have all cell membranes. Okay, and now that cell membranes will not become strong if we don't have a very good cellular respiration. So that was aerobic cellular respiration for all living organisms. Okay, but now we are talking to anaerobic, the opposite. Okay, like that, which is the anaerobic. There's a word an, huh? An anaerobic, Anaerobic process of cellular respiration. Okay, anaerobic process cellular respiration, there it starts also with glycolysis. Everything will start with glycolysis, okay? You understand, my friend? Class, it always starts with glycolysis, whether it is an anaerobic cellular respiration or aerobic respiration, cellular respiration, it will start with glycolysis, okay? Like that, and it will break to glucose, and after that it will separate it is going to the the cellular respiration by oxygen and cellular respiration without oxygen, okay, something like that. So we are going here on the other side, cellular respiration without oxygen, it will produce lactate and lactic acid, okay, like that. I will show you the drawing or the schematic diagram so that to understand further, right? Because we cannot understand further if we will not draw or we will not sketch the flow for this, okay? But to give you an idea, it's going to be like this. The application of this anaerobic process is that it is applied in all organisms, okay? Or organisms and bacteria are used to convert energy, okay? Bacteria or microorganisms are used to convert energy without the presence of oxygen, something like that. Okay, for example, animal manure, the fertilizer, like the wastewater, bisolids, and the food waste. So those are having the anaerobic process. Aside from the person, we have also an anaerobic process through that. Okay, so those are the applications. Okay, like that. So very helpful also. Okay, since we know that the plants need fertilizers to grow, and then if they cannot grow properly, then we cannot get food for us. Okay, to feed us. Okay, it needs fertilizers and animal manure, right? Something like that. So the anaerobic process is useful to that. So bacteria are used to break down those substances into energy, something like that. Okay. So without oxygen. So imagine we can survive without oxygen, right? Some bacteria. Okay, but not all. I think so. If others cannot survive also without oxygen. So there are two types of bacteria, right? That's why anaerobic. So that's why, okay, very clear. Now we will draw now the schematic diagram or the flow chart so we can understand properly. Okay. But if we speak about anaerobic process, still we have the pyruvate as our backbone for that. Okay, pyruvate meaning three atoms of carbon, four atoms of hydrogen, and three atoms of oxygen. So these are our backbone, whether we will produce the anaerobic process or aerobic process. So this is in the center. Okay, so still the same, it will produce pyruvate because this is from glycolysis. Okay, the glycolysis will produce pyruvate. Okay, two pyruvates, and this pyruvate will be doing the process of this lactic acid and the other one also for the cellular respiration that will produce there in the four processes that discussed a while ago, right? So it will split something like that. So, but my point is the two processes, whether anaerobic or aerobic, will use the pyruvate. Okay, that's the point because it is really the product of glycolysis to produce two pyruvates when the glucose will be break into two parts. Let's understand my English. I cannot show you in paper because my Scotch papers are too much discussion. Okay, full back to back had condition. Wow. Ganon. See? An evidence that I'm writing too much. Okay? Something like that. Okay? By the way, I love to blab or to live stream today because I don't want to sit after eating. Okay? Because, it cannot produce proper digestion if after eating you will sit immediately. That's my SOP, safe operating procedure. Even before, after eating, I will never sit immediately. Okay, I'll just stand, walking a little bit, stand, walking a little bit, so that there will be a proper circulation of my blood and at the same time proper digestion of the food. Because if you sit immediately, okay, they cannot properly do their job there. Okay, and then there's a tendency that you could develop a belly fat. Belly fat, okay, due to the bile acids, okay, produced by the liver through cholesterol content, something like that. Okay, so be careful for that also. Do not sit immediately, because your stomach will become big also in the future if you keep on sitting after eating. Okay, just 5 to 10 minutes, stand up. 5 to 10 minutes walking the floor or anything you do. Okay, and then after that you can sit. Okay, you can eat while sitting, but after eating you will not sit. That's my point. Okay, for proper digestion. Okay, like this. Going back. Okay, so it's easy pala to understand. The only difficult really is the molecular structural formula and their functions that involves terminologies that are very deep. Very deep. We did not encounter in our elementary education. We did not encounter in our high school education. For example, the acetyl CoA. I never encountered it in my high school education in my college. Okay, I encountered that just now because I researched and explored. Okay, because I'm very curious regarding the energy we produced. Where did we obtain our energy? Something like that. Okay, I need to explore more, and then I discover the chemical composition, something like that. Okay, so acetyl CoA is very useful talaga siya to give us energy. Okay, and then we produce the adenosine triphosphate. This is the main source of energy of all living organisms. Okay, so in short, we cannot survive without the adenosine triphosphate because that is the source of carbohydrates and sugar we intake in our body. The glucoses, the fructoses, the lactoses, the maltoses. Okay, since we need sugars in our body to give us energy. Okay, that's their main function. Okay, like that. And some also carbon and hydrogens also will be used up also in the developing steroid hormones, fats, phospholipids, something like that. So there's a relation also. Okay, so because those macromolecules have carbon and hydrogen contents also, they cannot survive without carbon, without hydrogen. Okay, and the protein also cannot survive without carbon, without hydrogen in the backbone, right? In the protein synthesis, okay, in the formula of the protein, something like that. So they are related with each other. Then if you try to discuss, you can connect those macromolecules, okay? Because they are really related. Like this, our topic is cellular respiration. And it could not be denied. I can talk to sugar, something like that. I can talk to phospholipids. Because that is to back up the theory behind so that you can understand really the specific topic of this moment in time. Wow. So, okay, ganon. Okay. But I have to write the formula first so that we can understand. Okay. So breaking of glucose. Okay. Breaking, meaning if there are 6 carbon atoms, if they break into 2 into half, so there are 3 carbons on the left and 3 carbons on the other side. I will just speak, I'll never put, because you can understand my English, right? So after breaking that one, these 3 carbons will produce the phosphoglyceride, something like that, okay? The phosphoglyceride, the 3 carbons and the phosphate attached on it, okay? So this phosphate coming from the, adenosine phosphate, something like that, okay? and then the phosphate group. Okay, and then the other side also, 3 carbons plus phosphate also, right? Because they are breaking into half. Okay, so satu pa, CCCP, something like that. Okay, so phosphoglyceride, something like that. Okay, why glyceride, Sotero? Because there are only 3 carbon, if you can remember, and then this is still a sugar. Okay, so glyceride, phosphoglyceride, or PGAL, just abbreviated as a PGAL. After breaking the glucoses, you will produce a PGAL. So what is PGAL? Phosphoglyceride, meaning the phosphate group is attached on 1 carbon of those glyceraldehyde, which is a monosugar in the aldoses family. So since there are 3 carbons, trioses, right? So we'll never use the formula of aldehyde for this, but use the phosphate attached to it like this. Since we are not concerned with the hydrogen here because it's breaking, right? We are not concentrating, so we don't illustrate anymore the hydrogen in this situation, okay? Because we never touch the hydrogen because it's breaking, okay? So only the phosphorus will be attached to the 3 carbons, and the left also have 3 carbons and the phosphorus, something like that. So break na siya. So after that, so there will be it will produce an energy, the adenosine triphosphate, 2 ATP in the left and 2 ATP also in the right, something like that. Okay, and then it will produce another components, which is the NAD, okay, and the NADH. The NAD will oxidize to NADH like this, and the NAD will oxidize to NADH, something like that. Okay, so in short, after that reaction there, it will produce a pyruvate. Okay, pyruvate in the left, pyruvate in the right. Okay, these 2 pyruvates, regardless of any order, in any particular order, these are reused in the aerobic process of cellular respiration and anaerobic cellular respiration or aerobic. So, okay, so we will touch now in aerobic because we're finished with the aerobic, right? The pyruvate we will use. So we will start also with pyruvate. Okay, now I don't have the chemical reaction is easy to understand, right? But oh my gosh, always talking, you cannot understand without showing, right? Okay, so I need to illustrate the anaerobic process. Okay, charots. Okay. Basta mo ano siya, mo-oxidize by NADH. Ganun. Gusto mo ng NADH pero NADH, ano siya, nicotinamide adenine dinucleotide is the definition of this NAD. NADH meaning the H is just hydrogen attached because it is a electron acceptor. It will accept the positive charge that the NAD will give, will donate. This nicotinamide, okay, adenine, which NAD plus there's a charge positive. Okay, so during the reaction, he will give his charge to this or the biological engineering, something like that, and then you employ the biotechnology, and then in there also you apply the genetic engineering. So I think there's more knowledge compared to the chemical engineers because the chemical engineers are focusing on the process. Really, it's very boring process. You have to or the laboratory, but actually chemical engineers are not really for the laboratory. That's only partial application for the chemical engineers. The laboratory specific are the chemists, the BS chemistry graduates. Okay, it's not really the chemical engineers because chemical engineers are for the process really. But the point there, chemical engineers know how to conduct laboratory tests. That's why chemical products company, okay, hire chemical engineers also apart from BS chemists, okay, or chemists, okay, like that. But chemical engineers are really designed to work in a process, in a field, okay, like this, to study the process A, process B, process C, process D, and you have to analyze the raw materials, the behavior of those raw materials in conjunction, how they behave to the parameters, for example, temperature, pressure, and then the volume, the volumetric flow rate, okay, how they go on the lines to lines, so that there will be no leakages somewhere along the valves. Okay, there's a lot of valves connection like this, right? In the flanges, and if there are leakages, and then there's something wrong with the behavior and the movement of that fluid. It could be a gas, okay, or a liquid, whatever. So you have to analyze the volumetric flow rate. Okay, you have to adjust and compute something like this. Okay, using the overall material balance. Okay, and applying also some formulas, the basic formulas, density equals mass per volume, blah, 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 so and so. You could also apply thermodynamics there, something like that. Okay, and then the physics also will be applied because kinetic energy always like that. Okay, because that's motion. Okay, because we incorporate the motion, and then we will apply also that how it behaves in ideal state if it is a gas. Ideal gas law will be applied also. Okay, how they behave, and then the standard atmospheric pressure, blah blah blah. Okay, at certain temperature and at a certain pressure at STP, and ganon. Okay, so process A, process B, process D. Okay, in safety, there will be an additional. We study that also, but in safety, we will concern with the injury of the people, if the lines will be damaged, will explode, or there will be a loss of the containment of the vessel will, will explode. So that's the purpose of those safety, process safety engineers. That's why mostly process safety engineers are chemical engineers because they know the process, something like that. Okay, the different physical and chemical separation processes. Because mechanical engineers are only focusing on the functions and operations of the equipment specifically. Okay, they don't know how the raw materials, how they behave, how they react. And it's in there that poses the worst situation. If they will react vigorously, it will explode, something like that. Okay, so it's no longer mechanical, right? It's more on chemical, something like that. That's why the hard process safety engineer, mostly chemical engineers, okay, especially in petrochemical industry, petroleum industry. So there are also mechanical engineers, okay, but in this field, they prefer chemical engineers, okay, for process safety engineers, okay, like that. Okay, you have to, of course, in process, you have to study first the process, what process you are in, to understand first before doing the HAZOP, what we call that HAZOP, right? Hazards operability study, okay, so that we can prevent catastrophic events from happening, okay, like this. Okay, so why we go there? Because I'm talking with biochemical engineers, right, in relation to the biological process. Going back to to pyruvic. Okay, my gosh, reaction. Okay, pyruvic. How can I put? See, I think this is also that was the difference between for all safety professionals. Listen to me right now. Oh, hello, all safety professionals all over the world. It is important to classify okay, the relevance of a risk assessment compared to the relevance of the job hazards analysis and the relevance to the HAZOP. Okay, the HAZOP construction industry. Okay, the HAZOP is for operational industries or manufacturing industries that produces products, meaning it's running already. Okay, so we cannot do HAZOP in the still going, the building is being constructed. No, we can only do the risk assessment and job hazards analysis, more on actions of the people. But regarding the process, it's HAZOP, hazards operability study, equivalent to risk assessment and job hazards analysis in construction industry, something like that. That's why they hired mostly engineers for that. Okay, for the for the manufacturing industry. Okay, because engineers have in-depth knowledge on the mathematics, calculations, process, science, like that. Okay, and we have extensive study for that for 5 years. Okay, that's why they prefer to hard a chemical engineer for process safety engineer, something like that, or for safety in manufacturing industry, something like that. Okay, But if you're very intelligent, even if you are not an engineering graduate, and then you are very, resourceful, you explore, you research, and through your experience, you can act also as a process engineer. There's no problem for that. Okay, it's depend on you how you do your task in the future, something like this. Okay. Right. So we have pyruvate. Okay. Pyruvate is really. Maybe you will think, so Sotero, how can we buy pyruvate? You will never buy pyruvate in the market, huh? It's inside our body. Okay? Very clear after the glycolysis process, the breaking down of glucose. Maybe you will, sir, can we buy pyruvate in the market? No, it's not. There's no such like that in the market. You can buy the Bacala or whatever. No, it's not being sold in the market. Okay? It's a functional chemical substance. Okay? Like this. So pyruvate is 3 atoms of carbon and 4 atoms of hydrogen and 3 atoms of oxygen. This is the formula, right? Okay. Pyruvate. Okay, remember this one, huh? Okay. Now we are in the anaerobic process. Anaerobic. English also matters, huh? If there's the word "an," meaning removal or without, like that, because matters a lot also. Okay. The prefixes of the words matters also. Okay. Dehydration, hydration, anaerobic, aerobic, like this. Okay. You have to understand also the words. You have to evaluate. Okay. Aerobic. Let's say process only. Okay. Process. Process meaning it involves a series of sequential steps to produce a final product, something like that. Sequential meaning systematic. Okay, it's not shortcut method. So there's a process. Then if you have four processes in one plant, then if you jump process A to process B, that is not a process. Because process is a systematic sequential steps to produce a product, something like, or an output. So if it is systematic and sequential steps, then you do shortcut, then it is not a process. It is an artificial process or fake process. From the word alone, right? Anaerobic process. Okay, this is the reaction. Okay. So this reaction will be like this. Okay, ganon. So let's put an arrow here. So pyruvate. Oh my gosh. Ganon siya. So plus. Ganon. NEDH. Okay. Ganon. Okay. Plus lactate. Okay. Lactate. I told you before the product is lactate, right? For anaerobic. Okay. Ganon. Like this. Muna siya. Okay. Anaerobic process, this one. It's very difficult, right? Me also find difficult. Because I'm not really a best biology. Okay. Though I found difficult on the terms sometimes. Okay. This one. But we have to understand only the mechanics, then interpret the behavior, then it's okay, right? Like this. Pyruvate plus NADH plus H is equals to NAD plus lactate. So the product is lactate, right? From pyruvate. Okay. And you'll ask, sir, how can you obtain the lactic acid? If you add water, lactic acid will be there. Okay, because water is a polar substance, high polarity, meaning conductive. Okay, so he will react to lactate to produce a lactic acid. Okay, so here there's no lactic acid yet because there's no water added. This is from pyruvate. Okay, so lactate. So I will discuss this one. So pyruvate C3H4O3 plus NADH, which is nicotinamide, nicotinamide, adenine, dinucleotide, okay, hydrogen. Or we can say hydrated, okay, nicotinamide, very difficult, nicotinamide, adenine, dinucleotide. See, very difficult, okay? Because I'm not studying biology, okay, but you know the reactions I can understand. Okay, but you know this adenine is very obvious coming from the nucleobase pair of the nucleic acid sequencing. You remember that one? Adenine, cytosine, uracil, guanine, and thymine. They will pair with each other to produce a genetic information so that after the genetic information, they will produce the gender of the baby. If it is a boy, if it's a girl, something like that in the DNA. Okay, so this is adenine. The same thing here. So meaning to say, it's used here in the pyruvate reaction. The adenine, only the adenine is used here. Adenine. Okay, because this is discussed in the nucleic acid. Remember that one. So this reaction still that adenine here. Ikawat niya kadiyot in order he can proceed the reaction here. Something like that. Okay, so there are also chemicals like that. Apart from that usage, they will be used also from another reactions. Okay, like that. They behave too much, right? Okay, so plus H, which is hydrogen. Okay, and then NAD plus, okay, charge plus, then lactate. Okay. Sa ganito ni siyang NAD, nicotinamide, adenine, dinucleotide. And then plus lactate. Okay? So ang lactate, I'll give you the formula of lactate. Charot eh. But basically, you know, I'll tell you something. Lactate will be the same, carbon and oxygen. 3 atoms of carbon, but the hydrogen is 6. Okay? Like this. And the oxygen is 3. Just remember that the carbon atoms and the oxygen of the pyruvate and the lactate are just the same. Only the hydrogen has difference. So that you can memorize faster. Okay, aside from breaking down the molecular formula structure, okay? Because it takes time to break the structure. And you write like this, blah, 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 so, and shared electrons, ganan. So it's very difficult. Not difficult, time-consuming, okay? Like that. So it's better to memorize things like this because it's not that really too much. Only 3 elements, right? CHO only, okay? So this is gonna be like this. Okay. Remember, the pyruvate and the lactate is an ester, okay, because it ends in ATE. Ester meaning it is produced by the combination of carboxylic acid and a long chain of alcohol. The product will be the ester and the water, something like that, okay? So, meaning in organic chemistry, this is still salt, like chromate, sulfate, phosphate, because it ends in ATE, okay? Or a polyatomic ion, okay? Remember oxalate, chromate, phosphate, sulfate, silicate, like this, bromate, iodate, okay? So those are polyatomic ion that has charges, ATE, and that's salts, okay? In organic chemistry, that is ester. The same, okay? But we are not really concerned with the charge because if you try to be strict, this pyruvate, this is not a pyruvate, this is a pyruvic acid. But in the biology, they are saying there, it's just used interchangeably. You know that? Pyruvate and pyruvic acid are just used interchangeably, meaning to say, if this is the pyruvate, this is the conjugate base of pyruvic acid, something like that. And then if you said lactate, it is a conjugate base of the lactic acid in the presence of water. Remember water, okay, because this is a esterification reaction, or shall we say hydration synthesis. There's an addition of water, okay? Again, addition of water is hydration synthesis, or it is an esterification reaction, something like that, okay? In organic chemistry, this is hydrolysis reaction, salt and water, okay? Always with water, okay? If you produce these salts and esters, okay? Remember, that's the clue, huh? If you have the salts and esters, it will be always partnered with water, okay? Or else, if there's no water, there's no acid, because of the H positive that will be trying to donate or to transfer there in the product, right? So it makes sense. Okay. Then, since in the biology or in the biochemistry, they are not really interested with a specific, uh, formula. They said there that pyruvate can be used also the same as pyruvic acid, like this, because this pyruvate is a conjugate base with pyruvic acid, the same as lactate. Okay, very clear. So, my gosh. Okay, meaning to say, if that is lactate, meaning it comes from lactic acid, right? But be careful, okay, because lactate is dangerous and lactic acid is not dangerous. If you talk about medical science, okay, right? So we produce lactic acid when we do the exercise, right? Okay, if you have, as I mentioned a while ago, if you do a lot of exercise, okay, if this, I will draw you a weightlifting like this, and you become tired. See, you become tired because the oxygen are gradually decreasing. You used up more the oxygen, and then your energy is keep on releasing. Okay, breaking down of those, energy keep releasing. So there will be a time come that your muscles will be tired and fatigued. Okay? Until such time, there will be no energy and ATP there, and you're very tired. You can no longer do, and you stop. So see, there's no such workout and activities that you will last for one day like this, because you need to rest, because that's the time no more energy released because of the anaerobic process cellular respiration, meaning there's no more oxygen that's circulated there because too much tired, he used up all the oxygen. Okay, cannot compensate already because use up oxygen, use up all energy, something like that. Okay, and that's the time you produce a lactic acid. Okay, so the sweat will come out like this, and in the sweat, the lactic acid is there. Okay, because the sweat is composed of mainly water, uric acid, electrolyte salts of any kind, plus the lactic acid. Okay, if you get a sample of sweat and you test that in the laboratory, you can identify the presence of lactic acid through qualitative chemistry analysis, not quantitative, because this is not amount. Only determining what kind of amount present in the sample. This is qualitative laboratory test. Sample, put in the test tube, and then do a series of laboratory tests and identify what elements and compound found on that sweat is WA80, and sure there will be a lactic acid there. Okay, so the lactic acid is one, the formula is here. So this lactic acid is not dangerous. Okay, this is a sign that the fat has been breakdown, the sugar has been breakdown, like that. Okay, so at that time you're very tired already and fatigued, you can no longer lift the weight because you're very tired anymore, excessive workout like that. By the time you like that, so your muscles will become acidic, the pH will rise, okay, because of too much, because there's a lot of lactic acid already produced. That's why it's acid, right? So basically, you undergo, your muscles have so many acids already, so it will become acidified. So you will develop a muscles acidosis. Charot. Grabe. Muscles acidosis. Okay? The process is known like that. So when we reach the muscle acidosis, that's the time you will stop doing the exercise because your muscles cannot handle anymore because the oxygen are already consumed, the energy is being used up, and your muscles will become fatigued, tired, like that. Okay? And lactic acid will be there, releasing in your skin, going to be lesser. Because this anaerobic process is can operate without the presence of oxygen. Okay? Like that. If you have disease like that, you will be admitted to the hospital. Critical disease, right? You need oxygen to breathe, right? So, of course, if you meaning you have lesser oxygen, okay? Because lactate will rise if there's an oxygen, less oxygen in our body. Because anaerobic, okay? So, this lactate will result to malfunctioning of the function of the organs. Okay, so if too much level of lactate in your medical result, meaning to say your disease will be severe. Okay, will be in trouble. Okay, so meaning to say you should be having a lower lactate level in your blood. Okay, if you have a very high lactate, meaning to say your main organs cannot function properly because this lactate will destroy the functions of your main organ. Okay? So like that. So it's very dangerous, this lactate really is. There's a level, the doctor will know that it's more than that. That's why if you have a critical disease, you need more oxygen, right? So if you need more oxygen, that's the time the lactate will become lesser also in your body. Because the more oxygen, the more this lactate will disappear. Okay? Because this is indirectly proportional with each other. You understand? The lesser oxygen, the lactate will be there. If more oxygen in your body, so the lactate will be lesser. That's why you don't have disease. Okay? Like that because you have lesser lactate only. The problem is if you have too much lactate in your body during the medical exam, then that's the time you cannot overcome, you cannot breathe properly, and you will die. Okay, because you lost oxygen already. Because the organs cannot function properly, right? It makes sense, right? Because, for example, if you have liver cancer and you wonder why you are admitted to the hospital and then your lungs also will be affected even though you have liver disease. So, meaning to say, this lactate affect the other main organ. Okay, you will go to different uh, bloodstream in the blood vessels connected to the organs. So just like my father, his disease is a liver cancer, okay? And then we wonder why his lungs is affected, too much blood, we pump the water in the hospital. So that's the purpose behind the lactate is rising, okay? Since the lactate is rising in your system, so that's very dangerous. And then the oxygen is becoming lesser because the water already there. So that's the time he loses breathing and he cannot sustain anymore and he died. Something like that. Okay, so this lactate is very dangerous. Okay, this one. Okay. But the lactic acid is okay because this is an acid that we release during exercise, jogging, biking, when we remove our sweat. Okay, so very funny, right? It belongs to the same compound, but the other one dangerous. Okay, because lactate is the conjugate base of lactic acid. Okay, and then why this one is bad and the other one is good? Something like that. You can understand sometimes also. That's why we need to do the exercise also. So that we remove the lactic acid and the lactate also will be digested. Okay? Because it will goes also to the lactic acid, right? Because it's the conjugate base, right? If you will not do the exercise also, and the lactic acid to disease, because that's the role of the lactate, it will destroy the function of those organs in your body. Really? My gosh, much. This is not the lactose, only lactate. That's different. Okay, that's sugar from the milk. There's no connection with this lactate. Only the same lactate, but it's not the sugar on the milk. It is lactate. Okay, the ester or the salt equivalent. Okay, so that's it. I will draw the reaction. Charot. Okay. Oh my gosh. Okay, lactate. My gosh. Mag-start ka sa lactic acid, right? Okay. Like, Very easy, man, to formulate because we have the lactic, right? Lactic acid C3H6O3. Okay, lactic acid. This is anaerobic process, all. This one I discussed. Because sweating, our anaerobic process is not aerobic. Because there's a, depletion of the oxygen in your body like this. Okay, lactic acid. Okay, plus lactic acid plus alcohol, right? OH is equals to lactate. Oh, gosh, huh? Okay. Ganon. Lactate plus water. So, in this case, you need trial and error, right? You cannot predict directly because you don't have an idea what is a lactate or about the formula. So since you have water that will donate one proton to this lactate, so you will just say C3H3 that will gain one electrons from the polar substance water because the positive proton of hydrogen will be transferred to the lactate. So it will become an acid, something like that. Okay, through theory only. So like this, and then you have to balance the equation like this. So meaning to say, there's a negative charge, 1. Okay, this one. Lactic acid plus hydroxyl group is equal to lactate plus water. And then if you reverse that one also, lactate plus water is equals to lactic acid plus OH negative. Meaning to say it is a reversible reaction. Reversible. It will go back to the natural and original reaction when you reverse the chemical reaction. Okay? Like this. So you reverse like this is just the same. Okay? So balance now. Always like to balance the chemical equation. Okay? Balance the chemical reaction. Okay. You balance now. So, ganon. So, carbon 3, ganon siya. This is a procedure every time. Carbon 3, hydrogen 6, 7. All. Hydrogen 7. Oxygen 4. Oxygen 4. See? Perfect. Balance immediately. Okay? So meaning to say this is a perfect reaction. Because you balance the chemical equation, all have the same number of atoms. So our hypothesis, which is an educated guess in scientific research, is assumed to be correct due to this chemical reaction we showed as a proof. Right? You remember the scientific research? If you do the research in science or any, if you have any experiment, you have to prove the validity of the reliability and correctness of your project in science. You have to have scientific research, right? You have to gather data, observations, inference, and after that you have a hypothesis. You have to guess what will happen, an assumption, and the last will be the conclusion, the final conclusion or the confirmation. So these are the steps that the scientists are using. Okay, all scientists are using these steps for scientific research. There's no also shortcut method for this. Okay, so after they concluded and confirm, not assume, finish only the assumption, confirm, that's that time they will create a formula and equation to the principle they discovered. For example, Albert Einstein, he discovered the theory of relativity, which is equals to energy is equals to the mass of the object times the speed of light at the vacuum, okay, raised to the power 2. Since C, speed of the light at the vacuum is equals to 3 times 10 to the 8 meter per second. That is how fast the speed of light really is in a vacuum. So Before Albert Einstein having that kind of equation, he undergo the steps of first gather the data, observation, inference, okay, go to the laboratory, and after that observe, and then have a guess, educated guess, a hypothesis, and after that keep on repeating, and it is correct now, and then you have the final conclusion, and now he can derive the formula out of it. Okay? It will never become a discovery if there's no formula and scientific law. Okay? The scientific law is always backed up with equation and formula. Okay? It will not end up with scientific theory and scientific principle alone. You have to back up it with scientific formula and scientific equation. That's why we have equations in chemistry, physics, okay, and even in mathematics. Okay, for example, Pythagoras theorem, you do also scientific research for that before obtaining the formula right triangle, right? C squared is equals to A squared plus B squared, and solving the value of hypotenuse. Blah, blah, blah. Okay, so those are scientists. Okay, it doesn't mean that scientists are only for science, also for other areas. Okay, we do the same thing. Okay, you understand the point? So this is also the same because I do it already also, and then I do the like this based on my knowledge, and then we come up with this reaction which is perfect. Okay. Uh, in the website I didn't found this also. Sometimes they will never also show. It's a problem, headache. Okay, sometimes. Okay, and then sometimes also they produce a wrong equation also. Really in internet charge to manganese charge like this, and sometimes I will just laugh and skip something like this wrong chemical. Maybe the author is not a chemical engineer or it's not a chemist. Maybe something like that because the compound he presented is having a wrong charge. Okay, so it will matter a lot during the chemical reaction because it will be used in the oxy redox, okay, reduction, oxidation reduction, because electrons and charges there are very important in the half reactions, right? You start with the half reactions, you balance the electrons, charges, blah blah blah, then you balance also the number of atoms later on. Okay, that's how tragic the oxidation and reduction really is. You need to balance the charges. Okay, in the half reaction, you have to divide that one, especially in acidic medium, in a basic medium, blah blah blah, so on so. Okay, like that. Okay, so perfect shock reaction. So sa to paingani pa siya. Okay, so this is really the real lactate. Okay, not this one because it's lactic acid, but the biology is using the same thing. If they say lactate, they use this formula. Parang ganon siya. Okay? They use interchangeably. Something like this. Okay? My gosh. Maybe it's not really that important, the reaction. That's why they keep on using the same, situation. It's not on the focus for their, topic, something like this. Because in chemistry, chemical engineering, you have to be specific regarding reactions. Maybe the biology part, they not really focus on the the details of the chemical reaction. That's why they use sometimes lactate as lactic acid. They use it as the same thing, just like the pyruvate and pyruvic acid, lactate and lactic acid. Okay, something like this. Okay, I already discussed to you the relevance and application in our lives regarding lactic acid, like this, blah, blah, blah. Okay, since pyruvate is the backbone, so all fermentation, fermentation in making wine, beer, liquor, whiskey, that's a process of fermentation there, okay? So they use the pyruvate as a backbone chemical to do that, okay? Like this. So they will use now the anaerobic process for that, okay, for the fermentation, especially the ethanol. Okay, it will be produced by the decarboxylation. Charot. Okay, like that. Masha, produce siyang ethanol. Okay, let's draw that equation. Okay, so do you understand? So there will be no beer, liquor, whiskey in the market if they will not use this pyruvate chemical to produce that one. Okay, and if there are no glucose reaction in the glycolysis, there's no also beer. It started all with glycolysis, as I said earlier, the breakdown of sugar and glucose and then converted to pyruvate and converted to alcohol. Okay, now it will not achieve for only one day. It will a long time. They will put in a barrel, let's say 30 days, 60 days, 80 days. The process took a long time before you can achieve a very good wine, liquor, and beer. It's not a magic, chunk, you can produce the beer. Chunk, it's fermentation properly, okay? Because there's a process for that. Exact proportion for the enzyme to catalyze the reaction, blah, 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 to produce this. It will take a long time. That's why they will put in a very big barrel in their company. And then if this is okay already, that the number of days required, then the taste will be okay now. They will release to the market, and that's the time they will pack, okay, in a bottle. Okay, that's not like soft drinks. Soft drinks is very easy to produce. Okay, they're just mixing and it will go to the bottling area and then it will produce and then tab QC like that. And the chemical engineers are computing the compositions and then the chemist will mix like that. Okay, the chemist cannot do the job without the chemical engineers approval and computation. Okay, that's the role of chemical engineers. Okay, and then they're waiting for the chemical engineers to submit the the proportions, the how many grams like this, how many like this, blah blah blah, and then before they mixed upside. Okay, and then we have log sheet like blah, blah, blah. And there we are working very fast. When the bottle will come there, we'll get there immediately and go to the gorilla test and so it's very quick. Okay, testing a lot. We have to test the the liquid, and we have to test if there are solids inside the soft drinks like that. We know that it's handling effect like this. The beverage product test. It's conduct one. Okay, the level of the sugar, the CO2. If the CO2 is very less, and then it will not taste good, something like that. Okay, and the water also will be proportioned for the CO2 like this, for the phosphoric acid content. Okay, like this. Okay, and then for the sodium benzoate, sodium benzoate preservative. Okay, I discussed it already, right? But they removed that sodium benzoate already, huh? In the Coca-Cola, something like that. Preservative, okay, for the soft drinks, carbonated drinks contains sodium benzoate, a preservative that is very dangerous to the health of the people. Okay, something like that. Okay, and then the concentrates. Then we mix everything in the tank there very fast. But the wine production is take a long time, the fermentation. Okay, it's not quick. Okay, there's a difference for the two processes. Sorry. Okay, and then we go to the reaction for that, for the fermentation. So this is, for example, this is glycolysis. What's one the glycolysis? How do they? Okay, no. Glycolysis. Okay, and then fermentation. Wow, fermentation here. Okay, like this. It produces also ATP every time because if there's no ATP, there's no energy and there's no glucose, right? Okay. Like this, and then this one will go here like this. That's a flow. That's only a flow, huh? And then we have here the ADP. You know the ADP? And ADP will catalyze to ATP. Adenosine, okay, adenosine dinucleotide phosphate, okay. I'm sorry, adenosine diphosphate only, sorry. Very difficult, right? Biology, okay. And then adenosine triphosphate, meaning to say the ADP will be converted to ATP, okay. So two components of ADP to two components of ATP, something like that, okay. Okay, like this. Because ATP is the source of the fermentation process for the ATP to ATP. Okay. And it will go like here, the pyruvate. Okay. And then pyruvate. And then the lactate here also. Lactate. So this will be used in fermentation. See?
Okay. In fermentation part, using pyruvate and the lactate. Okay. The glycolysis, start with the glycolysis. So when we say we cannot produce beer, wine, or if we will not do the glycolysis reaction. Okay. Like that. That's why the source are fruits, because fruits have sugars. Okay, so that will be converted to pyruvate and lactate later on to the process. Then through the process, there's a lot of catalysts and enzymes will act to speed up the reaction, of course. And I don't have to be detailed and discussing every time. This is only the direct principle behind in making wine, liquor, and whiskey. Okay, this is the process. Starting with the glycolysis, it will produce a phosphate, and this energy will be converted to pyruvate, and the pyruvate also will be converted to lactate, something like that. And the fermentation with there, bingo. After how many months, after how many years, the wine, the beer will be taste good. Okay, and this is for the wine industry, beer and wine industry, something like that. Okay. So in the center, before that also, we have the two I discussed, right? The NAD and the NDPH, just like a circle here. Okay, like this. This is the electron transfer chain, really. Oh my gosh, right? Here the NAD and the NADH partner together, right here. Here. Okay, here. Because this will be used in the electron transport chain. Okay, my gosh. Because you can never achieve pyruvate without using the NAD and ADH. NADH, okay, in the electron transport chain, okay, to produce the energy. Okay, so in the cells, okay, in the mitochondria, something like that. Okay, so you need to form the ATP from this NAD and ADH, something like that. Okay, as a result of the ADAPH and NADH, something like that. And then we convert it oxidized. It will be oxidized to ATP, something like that. Okay? Ganun siya. Look at this. Okay? Meaning to say, you cannot produce pyruvate without electron transport chain process. Okay, since pyruvate is very useful in the fermentation, so you have to understand also this electron transport chain, how they behave in the mitochondria, in the cell, like this, okay? So, because it needs ATP, which is the main source of energy, to be converted to pyruvate, right? So, it needs a transfer of electrons from these substances that will help these two, okay? Like this. Okay, so electron transfer process to form an ATP during the oxidation into the mitochondria. Okay, mitochondria is the powerhouse of the cell. Okay, so this is a cell like this. Meaning to say, these glycolyses are occurring in the cytoplasm. Okay, this is a cell and this is a cytoplasm. So the glycolysis is inside there. Okay, and there's electron chain, transport chain is occurring in the mitochondria. Okay, the powerhouse of the cell. Before we can achieve this pyruvate, like this. Too much process, right? Okay, so meaning to say, the anaerobic organisms are very important in this matter. Okay, electron transport chain, ganun. Okay, so pyruvate. Okay. So, okay, common man ang pyruvate sa duha. Okay? Very clear. Anybody? So, any questions so far regarding the anaerobic process like this and the anaerobic process cellular respiration? Okay. And then, let's go back to our assignment last Saturday. Okay, regarding uric acid. Okay. How can it produce the purine? Okay. I keep on thinking that one. I cannot sleep always thinking that one, really. I try to discover what's really the reaction, but it's not stated in the, in the website. It's not stated in the Wikipedia. It's not stated in the internet. Okay, I don't bring books here in biology also. Okay, and I don't have books here. Okay, so like this: uric acid, 5 atoms of carbon, 4 atoms of hydrogen, 4 atoms of nitrogen, and 3 atoms of oxygen. Let's consider pala, charot. Let's consider pala, salt, hydrolysis reaction, salt and water. Okay? Salt and water. Imagine this uric acid, this is not a salt. That's why it's an acid. So you have to plus this with OH. Okay? Ganon. To compensate the principle, base and acid as a product of hydrolysis, right? Okay. And then we have the salt plus the water. Now, where is the salt for this? The salt, of course, is the purine. Okay? Because this is the white crystalline substance that if you have higher levels of uric acid, this will exist in your bone, in the joint of your bones. You will experience an arthritis known as a gout, G-O-U-T, right? Because you keep on eating oily meat, oily hot dog like this, processed foods, dairy products like this, drinking too much alcoholic beverages. That's a source of rising your purine level and then eventually rising your uric acid in your medical exam result. Okay, especially beer have high purine. Okay, beer. Okay, so if you keep on drinking beer, there's a chance you have a high level of uric acid in your body. Okay, like this. So this is uric acid plus OH. This is a base to produce the purine, okay, which is the salt of the acid, the uric acid. So I know the formula of purine. So we are very lucky because I know the formula. Okay, C5H4N4. Okay, plus water. Ganon. Okay. So balance the chemical reaction. Okay? Because Saturday I found difficult, right? I used so many chemical reactions and I got confused also. Okay? So now let's review. It's an assignment. Ganon ako When I was in college, ganon talaga ako. Okay? If I cannot understand something, I need to dig it up and then until such time I can, I can solve it. I can really understand. I will never stop if I cannot solve it. Okay, I cannot understand the topic, something like that. That's why I always go to the library. So that atmosphere before flashback me now, okay, because I did not get that last Saturday, and I really find a way how to get this one. So I think like this. I use several reaction. What's the best reaction? And then it come up that this gonna be the perfect reaction for the uric acid. Let's balance the chemical equation. Okay, but I don't prepare, now just actual balancing the equation. I have now only an idea that's gonna be like this, like this. I will use this chemical reaction type, and then, but now, actual solving the chemical reaction. I don't prepare. Okay, so carbon 5. Okay, carbon 5. Okay, you count the hydrogen. Hydrogen is 5. Hydrogen is 6. See? Wala, hindi siya balance. Okay. Oxygen is giver. Oxygen. Okay. Oxygen is 1. Hindi siya balance. Okay. Nitrogen is 4. And nitrogen is 4 balance. Let's go first. What do you want? Oxygen first or hydrogen? Okay. I think any, right? We will start with oxygen because oxygen is the most abundant element in the planet Earth. Wow. Oxygen, okay, oxygen. So 3, 4, so 3 plus 1. Oxygen. So here, like this, 1. Okay, so let's just X. Okay, so X is equals to 4. Let X is the number of atoms of oxygen in the water molecule, so that you can understand, okay? Because every chemical balance equation, I'll never use trial and error method. I always use the principle of algebra to solve the number of atoms so that I can understand clearly. Okay, because others are doing the balancing by just trying 2, trying 3, trying 4 until such time it will become balanced. Me, I really use the principle of algebra that I let X, the number of this molecule, and then I solve that in linear equation so that I can obtain the number of atoms. That is the proper way of doing how to understand rather than too much trial, like start with 2, balance again, put 3, balance again, and then too much, too much trial, it will consume your paper, and then time will be consumed during the examination, something like this, okay? So we have to put 4 here. Okay, this is a technique, ha? Okay, so like this. So balance now the oxygen. So 4, so it's now balanced, okay? And now hydrogen, 5. Hydrogen here is 6 plus 4, 10. My goodness, 10. And then here is 4 plus 1. So let X the number of atoms of hydrogen in the hydroxyl molecule. Okay, something like that. So X. So linear equation, X is equals to 10 minus 4. So X is equals to 6 atoms of hydroxyl, and put that beside the hydroxyl as a coefficient. Okay, 6. And balance again. Okay, carbon 5. Hydrogen. Hydrogen now is how much? 4 plus 6, 10. Okay, the other side. Hydrogen is 6 plus 4, 10. Nitrogen 4. This is actual, ha? Because I didn't practice this one. This, okay. 4. Oxygen. Oh my God, I'm very, oxygen. Okay. 6. 6 plus 3 is 9. Okay. So 10 minus 4 is 6. Okay. So 6. So 6 plus 3 is 9. Okay. Charot. Oxygen here. 4. It's only 4. So we need to add 5. Okay. So 5 plus 5. So that is 5, 9. So that is oxygen 4. So 4x. Charot. Okay. 9. Okay. So 4x is equals to 9 over 4. Fraction. Don't be afraid with the fraction, huh? Because it's natural to have like that. Okay. C5H4 N4O3 is equals to 6OH negative C4N4 plus X is equals to 9,4 for the oxygen, right? So 9,4. So 9,4. Okay. And then H2O. Okay. Okay. So, ganon. So if we have the oxygen 9 here. Okay. And then your hydrogen will be 4. Okay. So, this is 4 here. Okay. And it cancels the denominator. Ay, chuba. Kaloka ka dyan. 4, 4. Cancels. So, balance again. Ay, for the 4th time, balance again. 5. Never surrender. Ganon talaga siya, ha? Because if you surrender, problem. N4, N4. Okay. Hydrogen. Ang hydrogen nato, kanahin mo na siyang 18. 18. Like this. 6 plus 4, 10. Okay. Oxygen is 9. And oxygen here is already 9. Gosh! 9. Hindi siya nababalance yung na lang. Yung, yung hydrogen. Kulang pa siya ng 8. Okay, so since kulang ng 8, so 6, 4, 6, like this. And then hydrogen is X. Okay. And then equal to how many hydrogen here? 9 plus 2, 18. 18 plus 4, 22. Charot. 6 plus 4, 10. Okay, so that is 4x. You need to 4x like this. Okay, so 4x equals to 22 minus 6, mga kapatid. Minus 12, 6, 16. Okay, 16 divided by 4, x is equals to 4. So put 4 here. Okay, again, this will be the sixth time. Wow, sixth time talaga, Jun. Yes, so 4C5, 4C5, H4, N4, O3 plus 6OH negative is equals to C5, H4, N4 plus 9 water. Okay? Now balance again. Okay, so carbon 20, carbon is 5. Okay, so x equals to 4. Okay, it's 4 here. Okay, so carbon 20, balance. Okay, grabe! Okay, hydrogen is 16 plus 6, 22. Hydrogen here is 18 plus 4 times 4, guys, how much? 16, right? 24. Oxygen plus 4 times 4, guys, how much? 16, right? 24. Oxygen is equals to 4 times 3, 12. 12 plus 6, 18. Oxygen, okay, 9 here, 18, so 9 like this. 9x. Okay, so x is equals to 2, so times 2. Wow. So 2, 8, C5, H4, N4 plus times 2 will be 18, H2O. Okay, like this. And now we have like this. Okay, so 8 times 5 is 40. 20, we need another 20 here. 20 plus 20, like equals to 40. So balance. Okay, so our problem already is hydrogen. Okay, so 18. Let's common denominator, my friend. Let's common denominator. So C5. Wow, algebra talaga. Let's common denominator. Simplify your reaction. If 4O3 plus 2, 3OH. Okay. 2C5. Ganon. In A4 plus. Okay. So, okay. 2, 9. 9H2. Ganon. So, carbon. This one. This is our final reaction. Charot. Grabe. Okay? Like that. So, too much reaction. Okay? So, it's gonna be, will reduce to purine and water. Okay? Like that. Okay? Okay? And then oxygen. Okay? Like this. And then if you undergo also the heat reaction, heat reaction, decomposition reaction, or let's say combustion, let's just try the combustion also. Maybe we can get the same answer. Another reaction. Let's see if we have the same result if you use the combustion. Okay, C5H4. Meaning to say, if you get the uric acid from your body, you have to burn the uric acid. Burn in the in the kettle, like that. C5H4 because there's a heat. Okay, O3 plus oxygen gas, right? Exothermic action. C5H4 N4 plus water. Okay. Let's balance the equation. Carbon 5, hydrogen 4. This is in terms of pyrolysis, ha? H4, H4, N4, N4. Okay. H4, H4, H6. Okay. Then 4x. So x equals to 6 divided by 4 equals 2, 2, 6, 2, 3 halves. Okay, like this. I'll put here 3 halves. Okay, like this. And then oxygen. So if there's a 3 halves there, you need to balance with 2 here. So it will become 3 C5H4N4 O3 plus O2 is equals to 2 C5H4N4 plus twice H2 like this. So 10, 10, 10, 10. Okay, plus CO2. Okay, okay, like that. So C5H4 and 4O3 plus O2 is equals to C5H4 and 4 plus CO2 plus H2O, like that. Okay, so carbon 5, carbon 5, 1. So 5, 6. So 5x, x equals to 6 over 5. And then 6 over 5 like that, 6 over 5 like that. Okay, so we have, so 6 C5H4 and 4O3 plus O2, and then since this is a 6 over 5, 5, so 5 C5H4 and 4 plus 5 CO2 plus 5 water, 30, 25, 30, 4, 4, okay, and then 12, and then 20 plus 10, 30. This one then 4, so 4, 6x, okay, is equals to 20 and 10, 30. So x is equals to 5, okay, so 5. So let's do this 30 C5 ganon H4 and 4 plus O3 plus O2 is equals to C5 ganon. So divided by 5, 6 ganon, 6. No, it's not the same. Okay, it's not the same. So meaning to say the the hydrolysis reaction, the combustion reaction is not really equivalent. Okay, by showing the reaction, by proving the reaction, it's not really, they don't have the same composition when we try to get the purine, because our purpose only is to get the purine, okay, in the reaction, because this is the dangerous one, okay, because this substance is found in many food we eat, okay, so this purine, something like that, okay. There's another equation also we try. Only one equation will balance, right? The salt and water will not balance, right? Oh no, the combustion will not balance, but the salt and water will balance. Okay, let's try the other one. Oxidation only. Direct oxidation reduction. Okay, in 4 or 3, then ganon. Dito siya. So equals to, so decomposition pala. Decomposition lang siya, okay? So C5H4 and 4 oxygen, okay? Okay, like this. So if we split the uric acid by decomposing it, the purine will separate from the oxygen gas, right? Correct? Since this is acid, where's the water now? It doesn't make sense, right? Because acid have water, right? Am I correct? Acid have always water. So where's the water? Let's just try this equation, huh? Because acid have water. It cannot be denied. So it should be in the reaction supposedly. But here, there's no water involved. And I'm asking only, where's the water? If we use the oxidation reduction method, let's balance. C5, C5, H4, N4, oxygen 3, and oxygen 2. Okay. Then 3 is equals to x. The x equals to 3 halves. Malakot ko sa fraction. Ayun, ang kalokat ng fraction. I don't like the fraction. Charot! Okay, ganon. So, it's gonna be 3 ganon and 4 O3. So, let's just assume that the water will be turned into vapor. It makes sense, right? The water in the uric acid turned into vapor and go to the atmosphere. That's why the urine will be left. It makes sense also, right? When you heat, the water will vaporize, right? What will remain is the solid substance. Chemistry, right? So it's gonna be valid, right? Okay. Because usually have water in the reaction. That's why it's acid, right? But when you heat this uric acid in a kettle, time will come that what will remain is the substance called purine. And this water will vaporize, right, by chemistry principle. Okay? It makes sense also. Okay? So, let's continue. It makes sense to O2. Okay? Let's just analyze, okay? Because it's not shown there in the website like this in Wikipedia or in the. I don't have books, as I told you earlier. I don't have books. Okay, so balance again. Carbon 15. Okay, like this. Okay, 3 halves like that. So cancel. So carbon 15. Carbon 10. Okay, hydrogen. Okay, oxygen 3. 2. So 3. Sorry po. Hindi po siya dito. Sorry ha. Sorry, because I misplaced. Again, I rewrite. So, when you do the balancing equation, don't be hurry also, ha? Because I'm in hurry, that's why. O3, okay, C5. I got 3 halves, right? 3 halves, or 3/2. So, improper fraction. Wow. If the numerator is higher than the denominator in algebra, that is called improper fraction, right? Okay. So, plus 2. The 3 halves, of course, it's in the side of oxygen because we let X the number of atoms in the side of oxygen, the side of the uric acid. Sorry. Okay. Ganon. So, ganoon siya. So, 2 moles of uric acid siya. Okay. 2 moles of uric acid is equal to 2 moles of purine. N4. And then, plus 3 moles of oxygen. Because it's balanced, guys. Maybe it will be balanced like this. We are very lucky if it will be balanced because we did not practice this one also. On the spot. I like on the spot. Okay. Carbon 10. Carbon 10. Look at, I have so many, assumptions. We try this reaction A, reaction B, reaction C, just to have a proof which is the best reaction suitable to produce this purine as a solid substance that is very dangerous to cause arthritis or gout, G-O-U-T, from excessive eating of oily food, drinking of too much alcoholic beverages. Okay, your uric acid will rise, something like that. So it is the purine is the indicator for that. It's not the uric acid, it's the purine because without purine, there will be no problem. Okay, like this. Okay. And this purine also is very far from uric acid. Guys, this is really the magic of chemistry because uric acid supposedly the name is urine. It's not purine. Then where this term came from? Really, right? And this formula of purine is coming from the uric acid. Supposedly it should be urate like this because it's related. Yeah, very unique, right? Compared to other chromic acid, of course, chromium because this is chromic acid. Sulfuric acid, of course, sulfate because it's sulfuric acid. But in this case, this is uric acid. Supposedly, that is urates. Supposedly, if you follow the rules. But in here, it's not. It's called, the salt, it's called purine. My gosh, ang lala. Okay? It doesn't follow the sequence of inorganic chemistry. Okay, like this. So going back to balancing liquid, that I found difficult also. Okay, to be honest. Okay, and then hydrogen. Be patient. Ganun talaga ang mga mga engineers. Charot! Hydrogen 8 plus 8. Charot lang ha. 8 is equals to, hydrogen is equals to 8. Nalingaw lamang ko. Okay, 8. Nitrogen. That matters the most. Nitrogen is 8. Nitrogen is 8. Hala! Nabalance siya. Perfect! Ito pala siya. This is the best reaction compared to the first one. Though the first one is okay, but I am not really confident because too much number of moles. I doubt why like this it turned 24. This one, I show you this is, the other one is very long, right? And then it doesn't make sense at all. It looks like very, confusing. We will use this one only, because it looks like logical to the concept of chemistry, okay? Okay, if we have the substance, liquid substance, we heat that in a kettle or any container. If we heat that, okay, time will come, all liquid of that acid will vaporize to the atmosphere. It makes sense, right? So what will be left is the solid substance of that particular compound or that particular acid. So in this case, we try to decompose the uric acid by means of heating in an exothermic reaction. Okay, when we heat that one, it will split into purine and oxygen gas, right? There's a process of oxygen because that's heating. You understand? Just like a combustion reaction. The only difference is there's no CO2 here because this is not petroleum or crude oil. So there's no presence of carbon dioxide because there's the presence of nitrogen. There's no rule in chemistry that if you try to combust a protein, even though it contains carbon, it will produce carbon dioxide. That's very principle, we should not memorize. Okay, in protein, if we try to do combustion by burning that protein, for example, it will not produce CO2 because the protein have a nitrogen bonded in the substance. So it will deviate the CO2 plus water in the product, even though we follow the same principle with combustion. Okay, now this is uric acid, the same principle with protein. It contains nitrogen. Okay, so it is not possible to produce a product carbon dioxide because this is not a carbohydrate, this is not a petroleum-based compound, and this is not a petroleum, and there is oil. So normally it will just split into purine and oxygen gas, and this oxygen will go to the atmosphere. And what happened to the water, Sir Sotero? The water, as you go heat, it will atmosphere also. So what will left is the substance, solid white substance called urine. And you balance the equation, it's perfectly. It does make sense by following the concept and the basic learning and foundation of decomposition chemical reaction. If you remember, we have 5 types of chemical reaction. Again, we have the chemical combination reaction, or double replacement reaction, single replacement or single displacement reaction, and we have the special reaction which we call as the combustion. Combustion chemical reaction is special. It deals only with carbon dioxide and water in the product. Okay, like this. And mostly CHO or CH are the compound to be added with oxygen to produce carbon dioxide and H2O. Okay, like that. So we are using the decomposition reaction, the splitting of molecules into several molecules during heating and exothermic reaction. something like that. So it will never hold through the hydrolysis, which is the reaction of salt and water. It will never hold through the combustion process, and it will never hold through also the esterification reaction. We tried a lot, right? So we see here, and it doesn't produce a logical chemical equation because it's very difficult to balance. Okay, like this, and then we come up with this. Okay, this is an analysis. This is sample of analysis, proving to a chemical reaction because, as I mentioned a while ago, it is not shown. It is not shown in the reference there, the reaction. I made my own reaction only. That's why. Okay, basing on learning and basic foundation of chemistry or chemical reactions. Okay. All chemical reactions will follow that one, the 5 chemical reactions. Except for organic chemistry is special. There's another reaction, leakage reaction, mga ganon, esterification reaction, ether reaction, ganon. Okay, there's different compared to inorganic. But in here, we consider uric acid as an inorganic compound pala siya. It makes sense or not? Let's think. Uric, right? Ammonia. Okay. I think so. Uric meaning your H4, ammonium, and H3, ammonia, something like that. So it's basically inorganic, right? Something like this. Okay. I think so. I think. Because there are also confusing compounds that you cannot really distinguish if it's inorganic or organic. All you have to do is to balance directly. It's not your intention to identify. Your intention only, your purpose only is to balance the chemical reaction, whether it is inorganic or organic, right? Okay, so that's your first intention. It's only being used so that you can understand what are the products to be to come up. That's why you need to identify with this inorganic, organic. Okay, but it's very difficult. Okay, like this. First time in my entire life. I'll never do this in my chemical engineering studies in university because this is, this is not really common. Okay, this purine, really. That's why I told you biology because purine is more on the health of the people. Okay, when we drink beverages, alcoholic beverages, the food we eat. So that's missing part of chemical engineers because we don't study that in our life, in our human anatomy, in our physiology. We don't study that. We're just studying the processes of the industrial processes like this. Okay, not on the life of the people. So that's why very interesting, right? The biochemistry and the biochemical engineering. Yeah. Apart from chemical engineering, biochemistry, then perfect. Additional learning. Ganon. Okay. Ganon na siya ha. So if somebody will ask you like this, so this is the reaction. Okay. The purine. Okay, this purine. So meaning to say, so you need 2 moles of uric acid before this purine will take effect as a dangerous substance in your body. You understand? Because if it will exist only as a 1 mole of uric acid, it will never be balanced to produce a purine. So it is unstable. So by basic principle, it will become a purine if you have 2 moles of uric acid to produce also a 2 moles of purine. That's the requirement. Okay, so if you have so many uric acid, let's say 8 moles of uric acid, then there is also 8 moles of urine. So that's the time your uric acid level in your system is very high. Because this is simplest. You start with the most minimal level of uric acid. This is ideal. Okay, so we still don't know how much is the level of your uric acid now in your inside of your body. So we know only that this is the reaction needed to produce purine in our body, which is dangerous if we can produce more than 2 moles of uric acid. Okay, so meaning to say this is natural process. This is not still dangerous. Okay, what is dangerous if it is more than 2 moles of uric acid that will produce more than 2 moles also of urine. Why I say that automatically? Because this moles is the basis of stoichiometric relationship. If you solve for the mass, okay, molecular weight, the density. So this will be included during the calculation. For example, how many compute like this? You can compute how many grams of purine like this. So that would be the basis. It's so many grams of purine in your body, so definitely dangerous. And we have the moles. And since we know that the unit of molecular weight is grams per gram mole, then you can compute the weight, okay, the molar mass. Okay, for example, you say how many grams of purine needed to produce uric acid 180 grams. Of course, you can solve that one because there's a number of moles and there is a molecular weight. So if you apply that in your body also, you can solve also how many grams of purine inside your body. If the doctor will say that this is the unit of the, the uric acid in your body is 110 milligram per deciliter. So using that units alone, combining that moles here in the chemical reaction, then you can solve the amount of purine existing in your body through that measurement obtained by the doctor. You understand? If you want really how many grams of urine there in your body, like that, okay? Because their unit is in terms of density. The doctors are using milligrams per deciliter. So that is density, right? Density mass over volume, right? And milligrams is grams, and deciliter is a volume. So density mass over volume. So they are using density as their unit as an indicator if your dosage and level of your uric acid is high or not. Just like same as cholesterol, okay, same as sugars, something like that, same as triglyceride. So if you really want to know the mass, not the density, then you can solve through this stoichiometric relationship. Since you know already the molecular weight of uric acid and you know the molecular weight of purine, which is the dangerous substance from uric acid if your body has more than on it. Okay, like this. But the body also want to know how many grams of urine you have in your body. If they receive already that is the results, and then they will accept like this. They will just determine the value that is higher and then it's higher also. Okay, because remember we have water, right? Water. There's also density of water. You understand? Density of water is equal to grams per ml. And we know acid has water. If you try to make in stoichiometric relationship, so there's a variation for the results of the doctor also because they're referring that as a density for the uric acid alone, I think so. But there's water there. Okay? Water. So 1 grams per ml plus the density of the uric acid. Okay? So total density of the uric acid compound or an acid is equals to the density of the water plus the density of the uric acid because uric acid has a water component in it. Maybe the doctor is referring only to the uric acid, not including the water like this. We don't know, okay? Or that is the total density already for the whole, including the water and the acid. Okay, like this. If you determine the density of urine, it's another story. Okay, because it's isolated, right? For example, if the doctor will give you isotonic, your level of uric acid is very high, 400 milligrams per deciliter. I know that it's not still the purine. Okay, it's still the uric acid. And we know the purine is the dangerous. It's not really the uric acid. Okay? Because if without purine, that is not dangerous by theory and principle. Okay? Our body creates a purine through the breakdown of chemical substances we eat. When we eat various substances in our body, and this purine is found in uric acid. Okay? Like that. Since it's very difficult to determine this urine alone by a solid substance, that's why they use uric acid as a reference alone. Okay, because it's very difficult to determine the solid substance in the readings in the laboratory. It will appear only the acid, not the solid. Now, the only way to understand how many purine is to do the chemical reaction. What is really the exact amount of purine through that uric acid that comes from the uric acid. We need to determine what is really the amount of purine substance, not the uric acid. You get the point? And in that, we can determine the amount through this chemical reaction. Use these chemical reactions. Now, if the doctor will say, your uric acid is 120 milligrams, use that as a basis of your calculation and do conversion factors involving the molecular weight and the number of moles, cancels there. Okay, and then you can obtain the amount of urine in your body, which is the dangerous substance found in uric acid. Okay? Like this. Of course, if you're not a chemical engineer, if you're not a chemist, you really say that uric acid is the danger. Okay? But since we know it's the urine is the danger, it's not the uric acid. Okay? Like this. Okay? Just like lactate or lactic acid. Okay? Sometimes we'll say the lactic acid is dangerous, but the lactic acid is the danger, not the lactic acid. Because lactic acid is sweet. SWEAT. Sweat. When you do the exercise. Okay? So this is very clear, huh? Oh my God, I found it difficult really for this. Uric acid chemical reaction. It takes me 1 hour to do that. Oh my gosh. It's happening. Guys, if you like this video, then you can comment, like, and share to anybody else if you want. If you don't want, it's okay. And then click the notification bell for some newest updates on I Am a Singer Eternal Voice. Focus on the coverage topics like occupational safety and health, science, and fitness, and medical science, and a little bit of mathematics, and a little bit of epidemiology. Wow, epidemiology. Okay, epidemiology, meaning we need to study a little bit with disease controls and prevention. Okay, like that through chemical reactions that will occur inside our body. Okay, like that. So we cannot do that without research, right? Okay, and readings like that, and then employing the knowledge of chemical reactions. Okay, how it behaves inside our body, in the physiological cycle inside our body, in the anatomy structure inside our body, which are the liver, the brain, the heart, like that, so that we become proactive to prevent diseases to come in ourselves. Especially if you're older, above 40 years old. Okay, that's the time diseases will come in, and then very expensive. The doctors are very expensive. And if we don't have money, problem. Okay, so it's better that we should prevent rather than cure and treatment. Okay, and I thank you. Thank you so much for watching this streaming. Again, God bless for supporting me, guys. Bye bye, everybody. Thank you. Charot.